So we're going to start with the Pro, Biosimilars Pro, and this is Dr. Brian Fagan. I think most of you will recognize him and know the name. Dr. Fagan completed his medical degree at the University of Western Ontario in London. That's Canada, not London, England. His postdoctoral training included residency. He's right next to me, making me nervous. He was a fellow in the Royal College of Physicians of Surgeons of Canada, and he holds membership in the Canadian and American Association of Gastroenterology, the American College of Gastroenterology, the College of Physicians and Surgeons of Ontario, and the Crohn's Colitis of Canada, and the European Crohn's and Colitis Organization. He's a recipient of the 2013 Senior Achievement Award from the Crohn's Colitis Foundation of America and the Dean's Award of Excellence in 2013 from the University of Western Ontario. I don't think we need to let the audience know. Brian has published extensively. He's been one of the leaders in the field. He's been a great friend and mentor to myself, and it's absolutely a pleasure to have him come and initiate our debates. Brian?
Well, thank you. Thanks, Scott. I guess I'm so glad that I don't have to debate Scott after that. Wow. So I find debates a bit of a challenge. You know, there's usually more heat than light from debates, but they are useful in one regard. These are my disclosures. So I'm going to start with the most important thing, how to win a debate. And those of you who have suffered through the last 5 years of watching television regarding your election, you know that there's some basic tricks here. First is suck up to your audience. I've never seen better looking people in all my years. You know, you're really exemplary. Second thing is denigrate your opponent. Now here's what bothered me. I mean, if I was debating Scott, this would be easy, but you know, it's Anita. I can't really do this. So I'm at a disadvantage. Come on, guys, I'm at a disadvantage already. And the third thing is distort the facts to make your case. And I think Scott alluded to alternative facts. I don't have to use any alternative facts here, and I'll explain why. So here are the key issues: that biosimilars are not generic drugs. They're not identical to the innovator. Why might it matter? And this is really the crunch issue, immunogenicity. And the immune system is exquisitely designed to recognize self from non-self. It's evolved over millions of years to do that simple function. And there's many determinants of sensitization versus tolerization, and sensitization has consequences. And this really comes down to one issue. And I'll use the term interchangeability, but I think a better term is non-medical switching. And that means switching from one drug to another when there isn't a medical indication. And that's a potential problem with proteins, and Anita's going to tell you all about that. And little is known about this in humans. So those are the key issues. So here's the first concept, that biosimilars are identical in terms of their primary structure to the innovator drug. So the amino acid sequence is the same. And if you've got good chemists, and most pharmaceutical companies have good chemists, then the secondary structure and the tertiary structure, which are really the three-dimensional shape of the molecule, should be the same as well. The protein should fold appropriately, and the disulfide bridges should be the same. So it really comes down to where does this generic issue come down to the fact that you can't create an identical molecule? Well, it's regard to the quaternary structure. And that's the carbohydrate groups which are tacked onto the protein post-translationally. And why can't we produce an identical product? Well, it's because the carbohydrate chains really are branched, and the way the carbohydrates, you can either go left or right at each branch, and there's an infinite number of potential branches. So the carbohydrate structure is complex, and it is part of the quaternary, determines the quaternary structure, which is what your T cells see when they decide to sensitize or tolerize. So it's really this variability in quaternary structure is really the key of this debate. So I'm going to talk about interchangeability mostly as the key issue and the risk of immunogenicity, but I will give a nod to extrapolation, which Anita will talk more about that, I'm sure. Well, here's where the problem starts. So if you accept that they're not generic drugs, how do they get approved? They get approved by PK study in 12 patients. If you show PK pharmacoequivalence, then that's good enough. So the regulatory authorities have decreed that that's not good enough here because although the amino acid sequence is the same, the drugs aren't identical because of that problem with the carbohydrate structure. So then they've said, well, we need clinical data in addition to, we're going to build the case on the basis of a variety of in vitro tests to show that the equivalent functionality, structure, chemistry, but we're going to require clinical studies. And the problem here is that statistically, you can't show that two things are identical, okay, just from first principles. So it's always a compromise. So in Scott's opening slides, I saw words like the same as, equal. These are not appropriate concepts. You have to decide whether you want equivalency, not equivalence, which means that the drug isn't better or it isn't worse. That's a two-sided test. Or non-inferiority, which is a more clinically appropriate situation. I don't care if the drug is better. I just want to make sure that it's not worse than. Well, this is just showing what the crunch question is, that if you're going to do this right, and I've given the example that you'll be most familiar with is a superiority study. So when we plan a superiority study, we figure out what the placebo rate is, we figure out what a clinically important difference is, and then we plug in the alpha and beta errors and behold a sample size. And so the example I've given here is a typical 15% delta for a superiority study, and you get a sample size of 320 patients. What happens if you want non-inferiority? Well, clinically important difference isn't what's relevant. It's what isn't important, a clinically insignificant difference. And we could play around with that, but for the most part, this is about half the clinically important difference. So 7.5%, and you can see what happens to your sample size, 1,500 patients. So non-inferiority studies are problematic because of large numbers are required. Well, this is the study that led to the approval of Inflectra, the biosimilar infliximab. And it's a non-inferiority, it actually was designed as an equivalency study, so two-sided testing, with an inferiority margin of 20%, which is very large. And this result, as a clinician, you can look at the bar graphs, these are ACR 20 scores, and one can argue about the clinical relevance of ACR 20 scores. But if you look at the two sets of bar graphs, to use a Scott analogy, well, there's no difference here, right, guys? And that's really what carried the day with the regulatory authorities. You can argue about the non-appropriate inferiority margin, but to speak to all the points that Anita's going to raise, I'll just remember, there's just one concept that I want you to remember. This drug was endorsed by your federal regulatory authority. It's approved drug. So the debate's over really at one level. So you can forget about substitution. Any pharmacist can prescribe this drug, and any plan can substitute it because the federal government has said that it's an approved drug. So, there can be a debate, but that's it. So the game is over as far as extrapolation and substitution. So I don't know why we're having this debate about those issues. Canada, Europe, every jurisdiction on the planet has approved this drug. So that's it as far as extrapolation and substitution. So let's talk about the really key issue, which is non-medical switching and the risk of immunogenicity. So here are the possible switching scenarios. So there's a one-way switch, then there's multiple switches with the same drug, and then really the kind of ultimate stress test is what happens when there's 5 biosimilars and you start switching back and forth between multiple biosimilars. That's really the issue around for the future regarding immunogenicity. Because in animals, if you want to break tolerance, one really good way is to take a protein that the animal's tolerized to and then just change the structure a little bit and then use interrupted exposures. And that's really a model for inducing immunogenicity, and that is really the fear that Anita's going to put into you. We've learned a lot about this, and this is not new concepts. So this is actually Peter Medawar. He and Sir Macfarlane Burnet in the 1940s won the Nobel Prize for their work on tolerization and sensitization, which ultimately led to the field of organ transplantation. And they identified that there were multiple factors which determined whether you'd sensitize or tolerize. And there's been many misconceptions about these years. We know that one of our antibodies is human, quotation marks, and therefore it doesn't sensitize, which is totally false. That's an alternative fact, which many of you have fallen victim of over the years. So cell line of origin is not a determinant of whether you sensitize or tolerize. And we've learned a lot about this ourselves the hard way. We kind of reinvented the wheel from 60 years ago. Things that were already known, we found out in the clinic. And this was really one of the first studies that really underscored that problem. This is Philippe Bert in New England Journal many years ago, showing that patients exposed to intermittently to infliximab, if they developed anti-drug antibodies, were less likely to remain in remission and more likely to have sensitization reactions, infusion reactions. And lo and behold, here's our human antibody, quotation marks, and it sensitizes too. This is a large cohort study published in JAMA some years ago in patients with rheumatoid arthritis. And you can see that about 25% of patients with exposed to adalimumab, despite the presence of methotrexate, will develop anti-drug antibodies over time, and in fact, they are more likely to fail treatment, having formed antibodies. So this is the premise, that if you have two molecules that aren't the same, a biosimilar and innovator, and you intermittently expose them, could you get sensitization? And you really need to assess that, because we don't want to sensitize patients to one or both drugs ultimately. And that really has not been evaluated. So we need well-designed switching study with multiple switches and assessment of immunogenicity and pharmacokinetics over at least 18 months. And we don't have any study for that. So that's the problem. So we need these types of studies. So I'm just going to take a minute to describe NorSwitch. And NorSwitch is a switching study. And it was designed by investigators in Norway. And they were given the challenge, so the Norwegian government said, you know what, we're non-medical switching everybody in the whole country because we're going to save a lot of money on infliximab. And the investigators said, well, we don't think that's a very good idea. We need to study this. And they said, okay, we'll give you 5 million kroners and you can study it. Have a good time, but we're switching. And so with the investigators given that problem, that's usually not the way we design clinical trials. So they said, well, we've got so many kroners and we've got to do this study. So let's take every indication possible, glom them together, take patients who are on stable infliximab for 6 months, and then switch them to either randomize them to switch to the innovator or continue infliximab. And we'll do a 1-year study, and the results will be based on failure. We'll use a composite outcome for all these conditions. So it's a kind of an unusual clinical trial, and it's a non-inferiority study. And you can see that, interestingly, the majority of the patients in this study had IBD. Somehow the rheumatoid arthritis patients got out of going into this. I'm told that it's because infliximab is not a preferred choice in rheumatoid arthritis in Norway. So nevertheless, we had over half of the patients with either ulcerative colitis or Crohn's disease. Now here's the results, and if you're not used to looking at these things, essentially with a non-inferiority design, what we're really interested is the margin of the confidence interval. And you can see the point estimate down at the bottom for the overall study, and it overlaps the line of 1. The point estimate favors actually infliximab, the point estimate, but the 95% confidence intervals cross the 0 identity line. So this is a positive study. Now what my esteemed opponent will point out is that in a subgroup analysis, that the Crohn's disease point estimate, although it does cross 0, is actually pretty far to the left-hand side of the figure. So therefore, if you're very simplistic about looking at clinical trials, you'd say that this shows that, it's not very good for Crohn's disease. Now that's a subgroup analysis, And as an epidemiologist, when we want to swear and use bad words, we start with the S word, subgroup analysis, okay? And so this is totally invalid to look at the subgroups. These weren't even pre-specified analysis. The aggregate was there a difference between the groups overall? So NorSwitch shows quite clearly that you can switch from innovator to biosimilar, but it's a one-directional switch. And you'd say, well, Brian, you told us you need multiple switches and you need to look at PK over time and all that true. And that's true. But does it matter? Is this really a provocative maneuver? Or have we already done this experiment? And here's what I will introduce you to, is the snowflake paradigm. So I told you that glycosylation occurs randomly, and it's uncontrollable. It's influenced by the culture conditions of the cells, and they're all different, and you can't replicate it. So that occurs with the innovator molecule as well. So you could imagine, at an extreme, that in a single vial of infliximab, there aren't 2 molecules that are the same. They'll have different glycosylation patterns. So we've been doing this for years. We've been doing this for 20 years. We've been exposing people to mixtures of drugs. So the 10 to 15% immunogenicity rate that we inherited with infliximab is due to this, and we have what we have. So there is no boogeyman out there that's going to cause trouble. So I think that's the issue, and I'll leave Nita to tell me why I'm wrong about that. So biosimilars cannot be considered generic drugs. Approvals have occurred for the first monoclonal antibody. Extrapolation and substitution are yesterday's issues. Interchangeability remains a critical issue in respect to immunogenicity, but will likely prove to be unimportant. Biosimilars are safe and effective alternatives to innovative products. Thank you.